You and I, from the young to the aged, this planet is home to all of us. There are countless risks in life, mainly notable in medical care, and in the face of global dangers, humanity has already accomplished tremendous things. But what if progress is reset?
We live in a world where bacterial infections have become fatal due to the emergence of multi-drug resistant strains.
Antibiotic resistance is now an urgent and severe global health matter.
As bacteria keep on developing and outwitting our antibiotics, treating infections becomes more challenging.
The World Health Organization projects a grim future. By 2050, antibiotic resistance could claim 10 million lives annually.
Thus, it needs a solution that not only treats an infection with resistant bacteria, but also prevents new resistances from emerging.
We are this year's iGEM Team Hamburg and hold a transformative vision that transcends all barriers.
Let us introduce transFERRITIN, because we want to transfer it in.
Our goal is to synthesize a drug delivery system by using a CPP-ferritin container that allows the specific introduction of antibiotic molecules into bacterial pathogens. Let's begin by delving into the remarkable potential of plant-based substances which will help us neutralize the resistant bacteria. We use the effect of synergy because the antimicrobial effect of these substances is much more effective in combination than individually. Therefore, it is important to keep these molecules together by encapsulating them inside a protein container. For that, we use ferritin, human heavy-chain ferritin. This iron storage protein is composed of 24 subunits.
Inside this complex is a cavity with a diameter of 8 nanometers, which is ideal to encapsulate our substances.
The uptake of ferritin into bacterial cells is limited. It needs a solution to amplify the process. We make use of universal keys.
The components responsible for facilitating the entry of our carrier into pathogenic bacteria are cell-penetrating peptides, short CPPs. We inserted one CPP at the end terminus of every ferritin subunit. We used 3 CPPs for 3 different constructs, TAT, R9 and R12. The advantage of CPPs, they can transport large molecules across bacterial membranes. Now that we have the cage, we need to load our cargo.
In order to get antibiotic molecules into our container, we make use of a special feature of the ferritin complex. At very acidic or very alkaline pH, the complex can disassemble into its subunits and reassemble at neutral pH. So our project idea was to disassemble the CPP-ferritin complex at a very acidic or alkaline pH, to then add the antibiotic compounds. And by restoring a neutral pH, the complex forms again, which then encloses our molecules.
For providing specificity, we use nanobodies, a smaller version of antibodies. We couple these to our ferritin to be able to target surface molecules on pathogens. For coupling the nanobodies, we make use of click chemistry. One part is the non-canonical amino acid transcyclooctaene lysine, short TCO, which will be incorporated in the ferritin's backbone. The nanobody will be modified with a tetrazine derivate. TCO and tetrazine will form a conjugate in a rapid reaction. Each subunit of ferritin has a barely conserved L-loop, which is located on the surface. We can easily modify this L-loop without changing the assembly of ferritin. In the assembly, the L-loops of 2 subunits are always next to each other. We can use this arrangement as a coupling site for the nanobody. In that L-loop, we are incorporating the amber codon. With a helper plasmid, we will incorporate a corresponding tRNA into the cell, which will be loaded with the TCO lysine with the according synthetase.
Then we add the modified nanobody. They will rapidly react, and the nanobody is covalently bound to the ferritin. With that, we have our transFERRITIN complex ready.
TransFERRITIN is now able to target specific pathogens, while commensal bacteria will be spared. The mechanism of action is simple. The nanobodies bind the pathogen, the CPPs penetrate the cell membrane, and the transFERRITIN complex enters. Inside the cell, the complex is degraded and the substances will be set free. And the pathogen is neutralized by the synergistically acting substances.
Let us have a look into our lab work. Our first goal was to obtain a plasmid without BSA1 and ZAP1 restriction sites to provide a plasmid for future iGEM teams that wish to proceed with our project or want to develop their own ferritin project. We used primers for a site-directed mutagenesis. After some former molecular biology steps, we sent the plasmids to sequencing. If the result came out positive, the process was repeated for the elimination of the second restriction recognition site. Our double digest and sequencing results showed that we successfully mutated. As a final product, we obtained the wild-type ferritin plasmid without ZAP1 and BSA1 restriction recognition sites, and therefore could be used for further modifications. To have a verification for a successful PCR mutation with CPPs, we assessed the transformed colonies through colony PCR. Colonies that resulted in a successful amplification were used for mini-prep and subsequently submitted for sequencing to verify the insertion of the CPPs into our plasmid. We first examined the PCR result by an agarose gel before performing a colony PCR. The successful sequence amplification was achieved for each CPP mutation. We managed to fuse each CPP with our plasmid. Now we needed to express and purify our proteins. First, the CPP-ferritin and wild-type ferritin proteins were expressed in E. coli. The second step was a simple heat and ammonium sulfate precipitation. Finally, an ion exchange chromatography and 2 runs of size exclusion chromatography were performed. In order to validate protein expression, we conducted a SDS page. Notably, we detected bands corresponding to wild-type ferritin and the respective CPP-ferritin construct in each lane. In a Western blot with an anti-ferritin antibody, we detected a protein in the TAT-ferritin and R9-ferritin lanes. A faint signal was also observed in the wild-type ferritin lane, so we successfully expressed our CPP-ferritin. The first step of purifying ferritin is a heat precipitation at 65 degrees Celsius, followed by ammonium sulfate precipitation. We checked the results of the purification with SDS page and Western blot again. After a Ponzo S staining, we could detect wild-type ferritin and the CPP-ferritin constructs. Ion exchange chromatography and subsequent SDS page analysis showed a purification of wild-type ferritin. The CPP-ferritin purification needs further process adaptation. We performed a size exclusion chromatography with wild-type ferritin and successfully purified this protein. To analyze the ferritin complex, we used negative staining after the purification. Under the microscope, ferritin appears as a symmetrical hollow sphere with a darker central cavity. We chose click chemistry to attach nanobodies to ferritin and created a binding site to provide specificity by introducing the amber codon. So we mutated the outward-facing lysine in the L-loop using quick-change mutagenesis and transformed our modified plasmid into E. coli. Sequencing of the plasmid with the amber codon proved the mutation. The introduced amber codon needs a helper plasmid to function as a binding site for the click chemistry. In this plasmid, we incorporate the corresponding tRNA as well as the amino-acyl tRNA synthetase using Golden Gate assembly. The TCO lysine that binds the nanobody is added separately and will get integrated to the tRNA. We proceeded with the colony PCR. Plasmid of positive clones were sent to sequencing.
What did we accomplish? We deleted the ZAP1 and BSA1 restriction recognition sites in the plasmid backbone, fused the CPP to the ferritin, expressed all CPP-ferritin constructs, and integrated the amber codon into the ferritin backbone. Furthermore, we achieved the purification of wild-type ferritin and did a negative staining.
iGEM is about generating knowledge that will be passed on through the generations. We would like to contribute to that thought. We were able to express, purify and image the wild-type ferritin. Since we eliminated the restriction sites, we can now contribute the whole plasmid containing the wild-type ferritin coding sequence. Future teams can directly transform that plasmid into E. coli and start their very own expression for producing a protein container for their usage. Ferritin displays the heart of our project, and we believe in it being the heart of many more ideas.
In the future, we will optimize the protocol for purifying the CPP-ferritin constructs. Moreover, we want to express the modified ferritin with incorporated TCO lysine for coupling the nanobody to the ferritin. With our transFERRITIN, we then will encapsulate the antimicrobic substances and test their efficiency against multi-resistant pathogens. Our hope is that our project will contribute to broadening the spectrum of antibacterial medicines that act primarily within the cell. We predict that by focusing on drug delivery, we will achieve higher efficiency and require less dosing, potentially saving resources and money. Furthermore, we can accomplish excellent selectivity and spare commensal bacteria that live in symbiosis with our body through precise penetration. These benefits outweigh the disadvantages of the favored antibiotic therapy, and we anticipate that transFERRITIN may give a therapeutic alternative to combat the problem of antibiotic resistance. Our contacts at Human Practices come from a wide range of backgrounds, reflecting the diversity of our project. We consulted various experts to gain insight into the current issue of antibiotics in a clinical perspective, including physicians. Many academia professors who have helped us shape our project fundamentally, industry representatives with whom we have collaborated, pharmaceutical industry representatives to start considering clinical approval, investors who have shared with us what is important in an investment, and a patient who has shared his experience of a past infection with a multi-resistant germ. After many discussions, we received valuable advice that we integrated into our project. This led us to apply for the Integrated Human Practices Award. For example, when we realized that CPPs were not specific, we incorporated the use of nanobodies into our project after talking with Alejandro Rojas Fernandez. Alejandro also suggested the use of click chemistry to fuse the nanobodies to the ferritin and create a binding site that would be more specific. We would like to thank all experts for their support.
However, science is incomplete without effective science communication. Our primary goal was to translate complex scientific theories, concepts, and discoveries into language accessible to all, regardless of their educational background. Our approach involved conducting a public survey and organizing workshops focusing on visual science communication and interdisciplinary collaboration. These initiatives helped us gain valuable insights into how to contribute effectively to the field. We spread this knowledge through workshops held at school, on Girls' Day, and during Hamburg's Family Day. We also had the privilege of representing our project at public events, including the German Unity Day and the peer-to-peer learning event, where we set up informative booths. Furthermore, we engage in academic scientific outreach by publishing an article in Biospectrum. Additionally, we participated in events like the Science Slam, the VAAM conference, and the European Meetup. As a culmination of our efforts, we even created a board game which was adapted as a video game about antibiotic resistances, a memory and a triplet board game. The reality of antibiotic resistances is a challenge we cannot evade. It's time to take responsibility. With our modular transport system, we offer an innovative solution for combating multi-resistant germs. The world of tomorrow is in all of our hands, yours and mine.
We would like to thank everyone from our team for spending so much time to shape our project with a lot of effort.
Also, a huge thank you to our sponsors. We wouldn't have come so far without them. Thank you!